See the beginning, role of fungi in urban forestry?
That's the one.
Beautiful. Okay, so quick disclaimer. Are we all on the same page? Literally? Same slide? Okay, quick disclaimer. No tree is safe, nothing is safe. So I'm going to talk a little bit about trees and fungi and about some fungi that are delicious edibles, choice edibles, but you must be 100% sure of what you are eating if you're going to eat it. And I'm not going to be able to teach that to you in an hour-long lecture. This is something that you have to do in your spare time. We have a lot of poisonous mushrooms in Ontario. This one right here, if you guys can see my my mouse, that's an Amanita virosa. So we're going to the next page here if we can. And looks absolutely stunning. And if you see it, it's actually a good thing for your trees because it's a mycorrhizal fungus. But bearing that in mind, it is not by any means an edible mushroom. So moving forward today, if I'm talking about risk, please keep that in mind, that all of these things are subjective and they're based on you and your experience. Okay, so moving right along, what's some of my background? McMaster University and Humber College, outdoor leadership skills instructor. You guys can get all that. If you have any more questions, I'm happy to talk about it. But at the end of the day, my life for the last 12, 13 years has revolved entirely around trees and mushrooms. My wife is incredibly patient with me because I don't get anywhere outside of those two realms. But I absolutely love them. So I'm happy to discuss this more. If you guys have any questions that I don't touch on the end of the day, or you found any questions about my credentials and how anybody can get them, it's a long road, but it's a very worthwhile one, and we're always looking for people in the industry because it is growing rapidly. Moving right along. So what are our goals today? We want to establish a foundation of knowledge. So I'm going in assuming that everybody out here has a bit of understanding on trees, on chloroplasts, photosynthesis, right? How plants grow, as well as a little bit of understanding around mushrooms. Addressing the urban ecosystem and the challenges for trees, because the urban ecosystem is not the same thing as the forest. The urban forest is not the same thing as a wetland. Trees have to deal with very, very different challenges in what is arguably the worst environment in the world for most life. Fungal ID and how trees cope with trees, or how trees cope with certain types of fungi, because fungal ID skills are something that we need to have a foundation in. Same with understanding the shape of a leaf, opposite and alternate leaf arrangements. It's a similar thing when it comes to fungi. Once you know a little bit of the basics, it's going to help you a lot with your field ID. Risks and rewards of the urban forest, because at the end of the day, everything we do, just like I was saying earlier, is risky. Risk is everywhere, and whether it's low or high, there's always risk. And sometimes taking the low risk path can have very, very low or weak rewards. And what are the costs of the urban forest? Because unfortunately, it costs a lot. It costs a lot to maintain trees. And from a private perspective and from a municipal perspective, I find most people don't actually have an understanding of what it costs and the technical skill required in actually managing an urban forest. And then how do you prevent common problems with your trees? So some things to look out for, and some things that you might see all the time, but you might not actually understand. So bearing that in mind, I think I can ask people to put their hands up. If we can get folks to put their hands up just to tell me who has a basic understanding of botany out there. So how to identify your trees, how to identify the differences between leaves, leaf margins, things like that. So I'm seeing about 9 people, 10 people. I expect more, but at the same time with the HNC, you know, there's a lot of birders out there, and there's a big difference between birds and botany, other than the bee. So bearing that in mind, we're going to keep moving forward. All right. And thanks very much for raising your hand and being involved in this. Okay, so moving right along. What are our urban challenges? Urban forest composed of 3 or 4 introduced species. We don't want that. We want an urban forest biodiverse in tree species and soil biota. So this is what people always get caught up on is, yes, we want to have a lot of different tree species, but what about the soil? Because at the end of the day, you are what you eat. And if you're eating junk food, you're not going to be in good shape. And a lot of the urban soils that we have out there are absolute junk. They are absolute junk for trees. Okay, so trees as ecosystems. To a microscopic organism, a tree is many, many things. And for fungi, they are living in, on, and around the trees, just like the insects, just like the tiny arachnids and the bacteria that live on and around the trees. And so moving forward, what I want to encourage everybody here to do when you're out and you're actually looking at your trees, and this is the neat thing, is you don't have to go to the forest for this. And this is a seed I want to plant for everybody, because I was speaking with Lou and Chris about doing a walk after this, whether it's next month or even later on in the season, because you can do a fungus walk any time of the year, because fungi is everywhere, all times of the year. You don't have to go out into the forest. There's a lot going on in the boulevard in front of your house. And sometimes that's even a little bit more fascinating, because you can see a tree that's really struggling with a bad environment and a bad diet. Microscopic organisms have to deal with a lot of things, and the bark is reflective of the forest surface. So I want to challenge you to start changing your perspective. Instead of looking at the trees just like a plant that's growing out of soil, I want you to look at the trees like its own ecosystem, with the bark acting like the top layer of soil, like the duff layer, and the wood underneath acting like the basic layers, the subterranean layers of soil that we all know about, but we very rarely interact with. Because yes, once you cut a tree open, it's completely exposed and we're used to it. However, when the tree is in its own environment, the purpose of that bark, the purpose of everything that the tree exists for, is to actually protect that wood so that it can live as long as possible. So when we're looking at animals, we're looking at how they get into trees, right? Because we've all seen squirrels that live into trees. How do they get there? Do they cause the decay, or are they responsible for decay? And one of the common issues that I've found in the past is a lot of clients ask me about carpenter ants, and they think carpenter ants cause problems. And the fact of the matter is that's not true. Carpenter ants are symptoms of a greater problem. They will show you something that's there. So when I walk onto a site and I'm looking at a tree, I'm not just looking at a tree, I'm trying to find out everything that's going on around it, because depending on where the fungi are, depending on where the insects are, I get a really good picture of what's really going on. Okay, so now what I'm going to try and do is, and I'm going to try and do this a couple times today. Can everybody see the Micro Capture Pro thing that I just brought up? Raise your hands if you can. Can anybody see that? Anything? Yes. Okay. Excellent. So here we're going to start right now. Look at this. We're actually looking at stuff under a microscope. This is one of the benefits of the webinar, I will say. So what we're looking at right now is, this is a piece of honey locust, Gladitsia tricanthos. So I'll move this all the way to the end. So this is the margin. So this is where the cambium would be, right? And so we'll move, zoom in a little bit. Now this tree had to come down because it was damaged. The client had damaged one of the main roots during construction, putting a post hole in. Now this is healthy wood, right? Now we're getting into, this is the heartwood here. And you see that crack along the side. This is where the decay started to move in. So the purpose of the bark is to actually prevent all of this decay from getting into the tree. So you guys can actually see all of the holes moving through. These are motorways for fungi, right? These are highways. And what the fungus actually does is break that wood down and start to live in there. It's an ecosystem. Now, sometimes it's a little bit uncomfortable to think about, because for the poor tree doesn't want anything to live there. The poor tree just wants to live itself. But it's always a constant balance. And so you'll see right along the crack right there what's going on. And then there's the healthy wood. Absolutely stunning. Okay, so we're going to go back to the PowerPoint. Wonderful. Just to give you guys a little bit of a taste of what I want to go into a little bit more detail. So trees as ecosystems. We just looked at the inside of the wood when it gets infected by decay. And right here on the outside, you've got, this is actually a silver maple. So silver maples, very, very common, especially in Hamilton and Burlington. They're one of our, I would say, our quintessential swamp species. Really like moist soils. They can sit in water for a little while and they grow relatively quickly. They're very robust. You can even plant them this is a caliper stalk, so what we would consider like 5 to 6 mil, a ball and burlap. If you look on the right side of the screen though, all of those black dots, those are actually fungal fruiting bodies we're going to look at a little bit closely, more closely later on in this webinar. And this is a species that was planted as part of an industrial park industrial park redesign, and these are all along the what they call in Australia the nature strip. So this is in Melbourne, and I was called out to this site in the middle of summer. Now, this is a classic example of right tree in the right place. Now, I know that there's a lot of controversy on native versus introduced and exotic species. Personally, for me, I'm not strictly a native person in the urban environment because the urban environment isn't a natural one. But if you're planting, you need to know what the heck you're planting, right? In this case, this is a silver maple. It had not received any water. I asked the contractor what their watering schedule is, and he just looked at me like I had 3 heads. If the tree isn't healthy, it's going to become more of an ecosystem to pathogens like fungi. Makes sense, right? So in this case, when I had asked, I was like, well, when was the last time you watered it? And he was like, well, mate, I don't know, like 3 weeks ago. Like, it's probably not that long. And for a tree, 3 weeks is a very, very long time. This whole subdivision, this whole redesign was full of silver maples that had no water. Trees that belong in swamps that had no water. Now, fortunately in North America, in Ontario, because they're native, they're used to the environment and our soils don't get nearly as dry as they do at the height of summer in Melbourne. You heap on top of it that it was terrible soil, and this is what you get is that decay, or not the decay, pardon me, but those pathogenic fungi on the side. And what we're seeing there with those black fruiting bodies, that's just the tip of the iceberg. That's, if you think about it, it's like a bulb where you've got the flower on the top, but you don't really know what's going on underneath. There's tons and tons of roots, or in this case with the fungi, it's hyphae, breaking down the cambium of the tree and degenerating it. So that tree is going to forever have problems. And I would be very surprised if they actually survived. Interestingly enough, one of the problems with this was that I had to go back to the client and just tell him that he needed to water the tree, which nobody likes paying money to find out that they just need to water the tree. But in most circumstances, and this is something we can all take with us, talking about the challenges of the urban ecosystem, one of the biggest problems I come across, trees aren't watered enough. Water your trees, especially when they're this small. When they're young, they need the water. Okay, here's another interesting one. This one's one of my favorites. This is in Ekaterinburg, Russia. So in the middle of the Ural Mountains. This is one of the largest cities in Russia that nobody's ever heard of. And these trees right here, these are poplars. So once again, part of the urban environment is managing the right trees in the right place. Now, from a professional perspective, this is a painfully hideous and terrible way to manage a tree, right? If you've ever seen a tree like this done, put your hand up just to give me an idea. So hands up if you've ever seen a tree. I would argue butchered, but also butchery is a really complex and technical approach. I wouldn't call this complex or technical, but it has its place. So the way that the environment works in Ekaterinburg is they're in the boreal forest. They do not have a lot of biodiversity there. And because they don't have a lot of biodiversity there, they have to deal with what they have, and they have to manage the trees that grow well. And one of the trees that grows really well in most environments, to be frank, is poplars. Now, poplars, though, they grow very quickly and they tend to fail. And so the way that the municipalities in parts of Russia deal with it is that they hack them back. They hack them back every couple of years. And that's why you get these atrocious cuts that aren't proper, because what will happen is in the following season, they'll grow out very quickly. They'll have that nice little ball on top. And for 2 years, they can leave it. And then they come back in the wintertime and they hack them all back. So the trees stay the right size. And to be frank, that's an excellent way to manage risk. It's not a great way to manage the health of the trees, but when you're dealing with the wrong tree, frankly, which is a poplar, I would say, in an urban environment, and it's the only choice that you have, other than if you see in the background, those look like dead spruces, those are tamaracks. You don't have a lot of options, so you do what you can with what you got. So I have to commend the Russians for that, because they've made lemonade out of those lemons. All right, so getting beyond that, friend and foe. We've got to look at fungi as complex organisms that play positive roles and negative roles. And it's really easy to kind of anthropomorphize these things. But at the end of the day, they just exist. That's all they do. They're just trying to live just like the trees are. All right, so what is fungus? It's a biological kingdom, right? So it's as different from plants as plants are from animals. Now, there is a lot of discussion on how closely related fungi are to animals versus how closely related fungi are to plants. One of the major differences is that fungus can't synthesize their own sugars like plants can. Plants and cyanobacteria are the only organisms in the world that can do that, with some very minimal exceptions. So now there are other things about the kingdom that people have mislabeled before, and I was talking to someone earlier about taxonomy. Slime molds and lichens are not fungi. They're closely related and they're fascinating, but they are not fungus. And we might learn more about them as time goes on, but one of the limits to our education and our learning of biology is the sort of funding that our academies get. And whether the academy can find a reason to do research or whether or not there's support from the private field, such as pharmaceuticals or small businesses or large businesses who can actually invest and find a reason to learn about these things. So the curiosities such as slime molds, they unfortunately get left behind. How do we define fungi? Mainly by the reproductive structures, and now a lot of the time by DNA. So there's a lot of issues in the fungal kingdom. We don't know that much about them. Okay, moving right along. So this right here, this is an interesting one that some of you may see sometimes growing on the underside of logs. It's actually not a fungus. I get this question from time to time. It's a slime mold called Hematrickia. And if you have a chance to look it up, it's really neat. And this was found in Hamilton, Ontario. So you can go out and find it. And what we're looking at there, so that is my pinky finger, my pinky thumb. And all of those little balls are fruiting bodies that at one point were part of a plasmodia. And that was an organism that was moving through the forest floor until it had enough energy to reproduce. Here is one that most folks should look at and be able to recognize. This is actually a very popular and highly sought after gourmet mushroom known as the chanterelle. Now, chanterelles are interesting because they're actually mycorrhizal fungi. Mycorrhizal fungi, of course, they're the fungi that combine with plant roots and are symbiotically, mutualistically beneficial. So they trade sugars with trees for phosphorus. And we'll go into that a little bit more, but this is an easy ID photo right here. I want you to look at that one, recognize it, because if you ever have a chance to see it in a farmer's market, pick it up. It's not very common. As commercially available as it is in Canada and the United States, there is no commercial growing for chanterelles or most mycorrhizal fungi for that matter, save for truffles. All right, so what are the parts of fungi? You've got the hyphae and the mycelium, right? So the hyphae are, if you think of them like the root hairs, right, that you would get on a tree. The mycelium is the whole body of root hairs. So if you look at one root hair, that's a root hair, and then you look at all of the roots, right? That's part of the one piece of the organ. So hyphae and mycelium are part of the same thing. And now when it comes to spores, that's what the fungi use to reproduce, and that's what sets them apart from plants, because they, save for the ferns and a few of the bryophytes, Hyphae, pardon me, fungi use spores to reproduce. So I've actually got a video here. I don't know if it's going to work, but before we go ahead, so you can see on the side here, put your hand up just to let me know if you guys can actually see my my mouse pointer move back and forth. Excellent. Okay, cool. Thank you very much. Look at this. This is really this is handy. I like that. Okay. You guys put your hands up. I know what the heck I'm doing, and we're all learning. Teachable moments. So on the left-hand side, you can see the really thin hairs of the hyphae, and then, like I was talking about a little bit with the highway system, you think of it like infrastructure, right? The older pieces get a little bit more robust, and we would actually call it chitinized, right? So they get more robust to protect themselves from the elements and from losing moisture, which is always a really important thing with most organisms. And all of this together is the mycelium, and the little individual parts, they're all hyphae. And you can see it takes on many different forms, and it can look very differently. One of the things that I've actually learned very recently, and it's not taught very often, you can identify specific species of fungi based on their hyphae. And not just with a microscope, you can actually do it macroscopically with the naked eye. It's just takes a lot of practice. And unfortunately, one of the challenges that we have is that we don't get a lot of exposure to hyphae because it's usually hiding under bark, or it's hiding in wood, or it's hiding in the soil. And by the time you dig it up and actually have a look at it, you've screwed it up so much it's unrecognizable. So looking at spores now, just to give you an idea here. So I've got a little bit of a video. This is a lobster mushroom. So a lobster mushroom is an ascomycotyl, and I know I'm getting a little into it here, an ascomycotyl fungus that preys on a basidiomycotyl fungus. So you see all these little orange dots up top? Those are all fungal fruiting bodies. And when I play, look at the right side of the screen, and you can actually see them all flying out of the fruiting body there. Those are tons and tons and tons of tiny spores. So we'll go right back to the beginning again, and you can see little bits. Put your hand up there if you guys can see the spores popping out. We'll do it one more time because I think it's super neat. My poor wife, when I had the microscope out on our kitchen table taking these, we were up at Algonquin, and typically I don't like to bring things home. You know, I leave only footprints, take only pictures, but I argue that because I'm taking slices for educational purposes, I can get away with it. If anybody wants to argue with me, I'm welcome to it. But I was taking this video at my kitchen table a couple of weeks ago, and I said to my wife, "Oh, Paulina, come here, look at this. Isn't that so neat?" And she was like, "Get that off my table. We're gonna eat on there later." She's a good sport. It's some of this stuff can be really gross for people, right? Not for me. I love this stuff, but for some people. All right, so going more into hyphae. Hyphae are like little straws, right? So they can be hollow, and it's the representation of the cell of the fungus. So every time that the cell reproduces, the hyphae start to grow. Now, depending on the type of fungus, they will actually make different connections, which we're not going to get too into because we'll get into the weeds. But you can see on the side here, so this is a eucalyptus, and this one we had removed on site and then come back a few days later to have a look at. And so you can see how much it had grown, and right along the margin here, this is actually where so the tree had already died previously, and that was one of the reasons we'd removed it. But the fungus was actually living on the outside, outside of the wood, between the bark and the actual wood, in the crack where the cambium used to be before the tree died. And coming back a few days later, it was remarkable to see that the fungus, which takes the path of least resistance, like most things do, had actually, instead of going just into the wood, had found that it was much easier to go up through the crack and around to the top of the wood. Really cool stuff. So there's another closer look at that same piece. So drink up, little buddies. Yeah, that poor tree though. One thing many people have probably heard here before, but I always like to say, especially when we're talking about urban forestry, if you look at a living tree and you look at a dead tree, the quote unquote living tree that's standing, that's full of foliage, is actually actually has less living cells pound for pound in it than a dead log on the floor of a forest, because that living tree is almost is protected against all of the other fungi and all of the other organisms out there. Whereas when it's dead and on the forest floor, it's being penetrated by insects, by salamanders and other small amphibians and reptiles and fungi. And what happens as a result is that dead log is actually full of life, more than it was when it was quote unquote alive. All right, so there's another shot of spores. Spore prints are one thing that you can actually do to identify specific fungi. And there are some fungus species out there where the living or the edible fungus is one spore color and the deadly poisonous fungus is another spore color. And there's only one way to identify the deadly poisonous versus the edible one, and that would be with the spore print. Fun fact. So make sure you do your research. Like I said earlier, I do not take any responsibility for what you put in your mouth. All right, so moving forward, many ways to disperse spores. This is another one. Puffballs are one that we get all over the place here in Ontario. I think this is a pear puffball. I can't remember the Latin off the top of my head, which is unfortunate. But you can see there are the spores coming out. Now, of course, if you get them early enough, they're, depending on the species of puffball, they're actually quite tasty. But you leave them long enough and there they are reproducing. Now, depending on the puffball and depending on the culture, some cultures in North America, First Nations cultures, especially in the north of Canada, the Dene, which you may know, some of you may know from the Northwest Territories and the Yukon, would actually use puffball spores as a coagulant to cure nosebleeds. So there you go. Once again, I do not, I don't advise that, but if you want to look into it, I fully encourage you. All right, so how are we going to identify fungi? And this is something I have spent hours and hours on. Some of my more professional lectures with the International Society of Arboriculture, we have workshops that are just on identifying decay fungi for a full hour. That's all we do, bang it out. And it can be really complicated. So this is all I want to do with everybody here today is I want to give you the tools so that when you go out, you can actually start to identify the different types of fruiting bodies, some of the characteristics that come with the different species. So understanding the phyla is a big one. So I had said earlier, Ascomycota and Basidiomycota, or Ascomycetes and Basidiomycetes. They are, if you think about it, the difference between vertebrate and invertebrate, right? We're animals, as lobsters are animals, as beetles are animals, as dogs are animals. But there's a very big difference between lobsters and beetles and dogs and humans. And that big difference is the spine, right? We are vertebrates, whereas lobsters and beetles are not. Both animals, but radically different in the way we reproduce and live and eat, and everything about a lot of our physiology is different. And the same goes for fungi. And it's the same goes for trees as well. When you're talking about conifers, right, versus flowering plants. There's a big difference between angiosperms and gymnosperms. And even though they're trees, right, and they have very similar physiologies in some senses, they're radically different in other senses. And this is how I want you to think of when you're looking at fungi, that if you're looking at an ascomycete versus a basidiomycete, that's a good way to start to learn the differences. And so if we look on the right-hand side here, those little orange pieces, those are fruiting bodies of the ascomycete Nectria cinnaberiana, which is a common ascomycete that we get all over the place here in Ontario. Some of you may be familiar with beech bark disease, which is one of the major causes of beech dieback in Ontario right now. And talk about a major urban forest concern. Beech bark disease is invasive, and it's what we would call a disease complex. So it's caused by an insect and a fungus, Nectria, right there. So those little orange fruiting bodies, are really indicative of the ascomycetes. Basidiomycetes generally, not always, but generally don't produce those types of fruiting bodies. So if you see something like that, that's a good indicator of what kind of fungus it is. And recognize those signs. So when you're looking at them, like a complex mushroom or decay, like a conch, like a Ganoderma fruiting body, which we'll go into a little bit more, those are generally basidiomycetes. So I like to put it, and this is a gross oversimplification, but I like to describe it as the vertebrates of the fungal kingdom, right? Basidiomycetes are a little bit more complicated. They might have a few different fruiting body forms. One of those fruiting body forms would be a complex mushroom that we all, that's iconic with fungi. And then most mycelia look the same. That actually needs to be updated because this is my discovery recently about the difference between different types of hyphae and mycelia and being able to identify. One of the ones to look at though here is those black lines, that's shoestring root rot, otherwise known as armillaria. So armillaria, very, very common. There are a lot of different types of armillaria around, very popular all throughout North America and Eastern Europe as an edible. Some people would say you should never eat it. Some people say it's okay, but you have to be careful. Other people swear by it and say it's like my father-in-law, say it's the best mushroom that you can ever get. At the end of the day, for trees, it's one of the worst mushrooms to ever find. And for me as an arborist, whether I'm dealing with a municipal site or a residential site, if I see it, and I actually was doing a consult earlier this month where I came across a client's house, a client's sugar maple that had these black shoestrings right there. That is an organ called the rhizomorph, and those rhizomorphs are indicative of armillaria, and armillaria is not a good fungus to have on your trees. It is a parasite and it is a decay fungus, so it is not nice. We're going to go further in. All right, so what are some of the basics when you're looking at your iconic mushroom, right? So you want to see if there's these things at the top where it says remnants. So it could be remnants of something called the universal veil, or remnants of the primordia when the mushroom started to form. So little dots, does it have little dots? Are they white? Are they yellow? Try and get an idea of the color. Does it have a cap? What shape is the cap? Because the shape of the cap can change dramatically, and we'll see this in photos later on, dramatically over the course of the day and the course of a couple days. So you could go out, see a mushroom today and go out tomorrow morning and it's a completely different looking mushroom, right? So the shape of the cap, how much the cap changes over a couple days. What do the gills look like? Do the gills connect to the stem? Do the gills sit flat? Do they dip down? Are they packed really tightly? Are they uniform? Do they alternate? All of these are major factors in the identification of a mushroom, which is exactly why I'm saying we're not going into that whole thing today, because it is an 8-hour course if you want to get into just the basics. It's basics. It's crazy. It's complicated. It's super cool. And it's a deep, deep rabbit hole that you'll never get to the bottom of, which is awesome because it keeps things from getting boring, right? So annulus and ring. So if you think about annulus is the proper term, but most people call it a ring. What does that look like? Does it exist, right? And that can be a major factor in the identification of some mushrooms. So to give you an example earlier, we looked at the agaricus and we looked at the destroying angel, and both of them had a ring, right? So that's not enough to actually identify it properly, but it is a significant factor for identification. Stipe and stalk. So what does it look like? Is it firm? Does it break apart in your hand? How does it break apart? Because even that can be an indicator of the species, because some will just flake apart. Some will actually stay together in strips. Does it come right out of the ground? Are there hyphae connected to the base? Because some of them, they'll come out of the ground like a cucumber. Other ones will rip out of the ground. Other ones will be connected to many, many other mushrooms from one spot at the base. And what those hyphae are connected to, that's what it's eating. So keep that in mind. When you're taking a mushroom out of the ground, if you're going to eat it or if you're just investigating it, and I always encourage, if you're ever taking a mushroom out of the ground, it's a nice thing to throw the cap into the woods afterwards, just to give the mushroom pardon the pun, but a tip of your cap to say thank you for letting me look at you and spread those spores a little bit for the sake of the mushroom. But when you take it out, look at what the hyphae are connected to. Are they attached to roots? Because if they're attached to roots, it's telling you what the thing is eating. So sometimes the fungus will be attached to root hair fibers, which means it's a mycorrhizal fungus. Sometimes it'll be attached to finger thick or wrist thick roots, which are structural roots. And then usually it's a decay fungus or a parasitic fungus. Sometimes those hyphae will be attached directly to wood, right? And usually you'll be able to gather that right away. But that should give you an example. And sometimes if it's coming right out of the soil, then it's just one of those subterranean fungi that lives in the soil under our feet. And it's eating all of the little bits and bobs that are in there that we don't think about, but they do. Well, I don't know if they think, but that's a discussion for another day. All right, moving right along. So this is the honey mushroom right there. So we were just talking about this when it came to identifying and recognizing an important urban forestry mushroom. If you don't remember any of the other mushrooms that I've talked about today, burn this one into your head. This is one of the most significant ones you'll come across, because if you have sugar maples on your property, if you have oaks on your property, in Burlington, we've got a few places where we're dealing with it, and it's particularly virulent. You can't get rid of it, right? It lives in the ground. Those gills, they produce billions upon billions of spores. Now, fortunately, lots of things eat those spores, right? Just like lots of animals eat the acorns that come out of an oak tree, but you only need one acorn to make a giant oak. You only need one successful spore to continue proliferating the honey mushroom, right? And so what will happen with this fungus is it will actually infect the roots of a tree, move in, start to parasitize those roots, sap the energy from the tree, kill the tree, and then use that dead tree and continue breaking down the wood and move through the soil to infect other trees in the forest. And so you can actually get an idea of where a honey mushroom begins in a forest based on where the forest actually dies back. And so if you're ever in a circumstance where you have one of these on your property, I highly suggest you call a professional, high quality arborist who knows what to do, because there are some species of trees worth planting in an area that has armillaria, and there's other ones that it's not worth it. Typically conifers, so your tamaracks, your spruces, and I'm talking about your native ones, because you want to give them every chance possible to resist this disease, or disease and decay. And ID points, right? So you're looking at the ring there, looking at the different pieces of color. So interestingly enough, and I love this about fungi, all of the mushroom that you're looking at, that's just hyphae piled on top of hyphae, piled on top of hyphae, piled on top of hyphae, so that it's so dense and so thick that it forms a robust mushroom. Or in the case of this cluster here, many robust mushrooms. The coloring is a nice indicator, and you can see I was talking about the gills, so it alternates a little bit. You've got a nice big gill, small gill, and then a tiny gill, and it does that all the way throughout. And that could be enough to differentiate between two different species of mushrooms, right? And they might be in the same genus, they might both be armillaria, but there are many different species of armillaria out there. Okay, moving right along. What else are we looking for? So we talked a little bit about mushrooms, but what happens when you're looking at a conch? And if somebody can just give me a shout, I just wanted to get an idea. How much time do I have here? Because I know I've been kind of spouting off and I've not been looking at the clock. I see it's 20 past 8. Do I have about 25 minutes left, 30 minutes?
Go as long as you want.
Brilliant. That's what I want to hear, Chris. All right. So what we've got right here is, so this is a Fomitopsis pinicola, another decay fruiting body. And you can see in the background, that's actually off of a sugar maple log. So like we were talking about earlier, 95% of that log is alive, arguably speaking. But what we're looking at here, it's a little bit different than your fungal, than your mushroom, right? So your conch fruiting bodies, your wood decay fruiting bodies have a little bit of a different structure. So what we're looking at here, we've got the pore layer. So all of those little things right there, and we'll look at it in a little bit under the microscope, actually in a moment, under the microscope, each and every one of those pores is producing spores, right? Spores come from pores. What you've got on the bottom, right, that's the final spore producing layer, and that's the newest. So some fungal fruiting bodies, especially in the world of decay fungi, can be perennial. So they can live year after year after year, which for a professional such as myself is really handy because I can walk onto a site, I can look at a fruiting body and say, oh, this thing's been here for 4 years. So know that there's 4 years of decay breaking down the interior of that tree. That's really important. And depending on the species of decay, I can actually get an idea of the biochemical makeup of the tree after the fungus has come in, because some fungi break down lignin, other fungi break down cellulose, which is a really critical part of trees. And we'll talk about that in a little bit more time. So finally, the last part, you guys see that part right here? This is one of my favorite jokes. My wife always rolls her eyes. So that part right there is called trauma, T-R-A-M-A, not the same kind of trauma that we all pick up from our parents. This is a little bit different, but it's more of those hyphae piled one on top of another, very, very thick, and it's very absorptive. Now, depending on the species, this one's a lot more robust, and this was at the right time of year, so you can see just how much water. It's like a bloody sponge. Look at that. Neato, eh? Okay, so here we've got a Ganoderma australis on what I believe is a Eucalyptus regnans. So lots of different types of Ganoderma, but this is a relatively, Ganodermas are a very common genus. And it's very important in urban forestry because Ganoderma is one of the more common decay fungi. And they're important to know and important to recognize because you'll see it often. One of the problems though is because it's relatively easy to identify, a lot of people misidentify other fruiting bodies as Ganoderma because it's the only name they know, right? Which is why sometimes a little bit of knowledge is worse than no knowledge. So if we look at this one right here, you can see it's kind of ballooned out. In the following year, that white margin on the top, that'll harden off, turn brown, but the bottom end will continue producing spores. And you can get an idea of the age. So you see it's a typical pen, which is maybe about, I don't know, 12, 13 centimeters long. You see the brown right there where my mouse pointer is, that would have been last year's fruiting body. That would have been as big as it got. And then this year, so this was taken in 2016, that would have been 2015, and then this is the year 2016. So now what we're going to do is go to Ganoderma fruiting body from good old North America. So we're going to switch back to the microscope. Here we go. All right, folks. So here we are at the top of a Ganoderma applanatum, otherwise known as artist conch, right? So you guys can see right here the margin. And now it's really interesting to see how hard it is. And this is one of the problems that we get in North America. One of the problems we get when it comes to control of fungi in the urban environment, because the reason this is so hard is because of this biochemical that fungus produce called chitin. Now, chitin is the same chemical that insects use to produce their exoskeletons. So if we actually produce something, and this is how a lot of biocontrols work, is meant to break down our cells, or break down the cells of a plant, or break down the cells of the fungus. And the easiest way to do it is to break down the cell wall, or to cause them to grow exponentially so that they die. In the case of chitin, if we created something that broke down the cell walls of fungi, it would also kill most insects. And that's why a lot of fungi are actually really, really difficult to control. So,
can I interrupt you for a second?
Yeah.
If you've got the microscope on a separate window, I think you need to share that window and stop sharing your slides. Because I don't think anybody can see the microscope.
Okay, hold on. So I'm sharing, new share. Thank you very much, Lou. All right, can you see it now?
Yes, can see that.
Brilliant. All right, let's go. So we're back to the Ganoderma. So I went through the whole landscape of the top of the Ganoderma. So this is the older stuff here, right? And this is the chitinous stuff. So this is the fruiting body that's really hardened off. And if we go further along the edge, so this is one of the margins of the years right there. So you can actually see where year 1 ended and year 2 began. And we'll go all the way to the very edge. And this is, you can see right nice, that nice white margin there. Oh, and that's the locust. That is what was left before I actually took the fungus off of the tree. Give me one moment here, folks. Just plug in my computer so it doesn't die. There we go. All right, now flipping right along to the back side here. Boom. Everybody see that? Hands up. These are your spore, or these are your pores where the spores come from, right? So just to give you a really good idea, these can actually be an identifier based on the species. Now, right there along the side, just to give you an idea of scale, that's the tip of a knife, right? That's my field knife. And you can see how small those spores actually are. So that knife, the tip right there is probably about 2 centimeters long. So look at how many are on there. Now, because Ganoderma is, oh, you can see, and there you go. So you can see the mark that I'm making, how gentle it took, like that took nothing at all. And I damaged those pores, which is where artist conch actually gets its name. Because if you get a fresh one, you can draw on it because those spores are actually really, really easy to mash out of shape. And it's one of the actual identifying features of Ganoderma applanatum. Very interesting stuff. So we'll move right along here. If you guys have any questions about it afterwards, let me know. And if you guys want more microscope stuff, also let me know, because this is something I've been playing with more and more. And the webinars, people seem to really enjoy it because it gives them a different perspective. Finally, just moving along, this is what the edge, the margin actually looks like. So there's no pores there, right? This is just there for growth. Because it's not at a straight angle, and you can tell I'm having a hard time focusing because it's curved, the fungus isn't going to produce any more spores coming from that spot. And in the following year, what would happen is that it would actually put an extra layer of spores on the bottom. Okay, pardon me, pores, not spores. Ugh, tongue-tied. Okay, so we're gonna go back to the PowerPoint slideshow, and we'll say thank you to Lou for for giving me a heads up on that one. Okay, so pathogenic fungi. They actively attack the living part of the tree. Once they're in, they're not coming out. And the difference between, there's a big difference between signs and symptoms, right? So a sign of a fungus is something like your hyphae, your fruiting body, rhizomorphs, spores. Those are signs. Symptoms are things like dieback, the bark flaking off, right? It's like, if I'm coughing, that's a symptom, but a sign of COVID is actually the actual viruses, or something along those lines. I'm not a human epidemiologist, so forgive me for all the professionals out there who are cringing at my terrible reference. One important thing to keep in mind, and I really try and hit this point home, a pathogen is something that attacks living tissue. Saprophytes attack dead tissue. So typically decay fungi, I wouldn't consider pathogenic. Now this is a very arguable, which is why I include this. This is another Ganoderma australis on another Eucalyptus regnans. As you can see, there's a lot of Ganoderma out there, or I just happen to find it. It's something that you can argue until the cows come home. Maybe by the time the year 3000 comes along, we'll have a decision. But until such times that happens, we're out to lunch. Other things to note, so in the left here, that's a Xylaria polymorpha fruiting body. That's an ascomycete, and there are factors that help you identify it as an ascomycete, which we may go into a little bit more detail, but I want to get to the end of my slides before 12:30 a.m. All right, so our substrate, what are we looking at? Substrate is whatever the fungus is growing in, right? So in the case of a pathogenic fungi, the substrate could be the roots, or it could be the cambium, or if it's a decay fungi, the substrate would be the wood. In the case of a mycorrhizal fungus, the substrate could be something such as the mulch right here. This is an interesting one that I like to refer to. So when you're talking about substrates and you're talking about mycorrhizal fungi, this is one of my favorite plants out there. And that's a tall order because I have a lot of favorite plants. Monotropa uniflora, otherwise known as Indian pipe. So you notice that it's white. This is a plant, but it's a plant that doesn't produce chloroplasts, right? it doesn't produce chloroplasts because it hijacks sugar from mycorrhizal fungi. It actually hooks into the web of mycorrhizal fungi in the forest floor. And this is a native Ontario plant and grows by stealing sugar from the mycorrhizae. Now, it's possible that it is a mutually beneficial plant, but as far as we know so far, scientifically, it is just a parasite of mycorrhizal fungus. And there's a microscope view of one of the leaves. Unfortunately, they don't last very long when you pick them, and I had taken a small cutting to get some photos under the microscope, but you can see there all of the cells and how empty they are. They're one of the few plants that I've ever seen that's, you know, you can kind of get a better picture of what's going on, but this is just white. And you hold it close enough to the light, it's actually clear. There's not, those leaves just exist. They don't exist to produce sugar, which is kind of redundant, right? Like, why would a leaf exist if it doesn't act as a solar cell? All right, so where are you gonna find fungus? There's a little bit of a digression there, but anyway, not important. Fungus grows on every part of the tree. So when you're in the urban environment, when you're looking at your tree, whether it's your magnolia out front or your Norway maple, your silver maple, your oak, you can look all over the tree. And when I'm dealing with clients, I typically go and I say, like, I can find something wrong with every tree, and it's true. The fact of the matter is some things are really wrong and important to address, and other things, they're not a big deal, right? And we all have our own problems. All of our trees have their own problems. The question is it going to affect the aesthetic value of the tree? Is it going to affect the valuable life of the tree? Is it going to introduce risk, and how much risk? Because there's a very big difference from, this is a dead limb and it's going to stay there for 7 years before it falls off, and this tree is experiencing advanced decay and is 75 feet tall and is going to fall onto a school. We'll see a picture of that a little later on. So where are you going to find the fungus? You're going to find it on the foliage. You're going to find it on the cambium. You're going to find it in the vascular system. Now, vascular system fungi, you're usually not going to see unless you're a professional such as myself, or you're a keener and you're doing your own pruning. And vascular stuff, vascular fungi, before you see the signs, you'll see the symptoms, right? So Dutch elm disease is a very, very common example. Sudden oak wilt, and this is one for everybody, to all the keeners out there. If you don't know what sudden oak wilt is, look it up. Look it up as soon as you get off here. It's a big problem. So Dutch elm disease can kill a mature elm in a season, and many of us have seen it, and many of us have dealt with the problems that come with that. And we're dealing with the same problem with emerald ash borer, right? When you have a monoculture and an invasive species comes in, it can really cause some devastating impacts, or have some devastating impacts. The vascular pathogens, they're particularly devastating because they can move through a tree faster than the tree's system of compartmentalization actually can react to it. Sudden oak wilt right now is in Detroit, in New York State, and Pennsylvania. It's right across the border. It may even be in Canada right now. If an oak tree if it contracts sudden oak wilt, it will die within a season. Doesn't matter if the tree's 30 centimeters in diameter and 25 feet tall, or 150 centimeters in diameter and 150 feet tall. Sudden oak wilt will kill a tree. Vascular pathogens, vascular fungi, are incredibly, incredibly virulent. They will wipe out a tree incredibly fast. Now, there are smaller, more common ones that are less particularly damaging to trees, such as verticillium wilt and Phytophthora. Phytophthora is not ideal, and that's a root rot that makes its way into the roots and then infects the whole vascular system. Verticillium can live in the wood, and when the tree gets weakened, it can actually take out entire branches or entire leaders and then sit in the tree and just wait. But Dutch elm disease, sudden oak wilt, those ones in particular, really particularly virulent vascular system pathogens. Cambium, those are your cankers, right? So many of us have seen like target canker on maples, which usually starts in the nurseries. Those are pathogens that only actually affect the cambium. So I was talking about Nectaria cinnamariana earlier, beech bark disease. That one only affects the cambium of the tree, only affects the cambium of the beech. The problem is if it reproduces so fast, it can wipe out enough of the cambium that it destroys the system of transport between the roots and the shoots at the top. And once that's done, the tree is toast. Whereas if you look at this image here, this is a very small, it's probably about 5 centimeter long and 2 centimeters wide, that fruiting body. I think that's on a sweet gum, a liquid ambar. And that's not enough to damage the tree. It's just gonna suck a little bit of energy out, right? It leaves an open wound, which is never pleasant. Anybody who's had an open wound would agree. Not that it's the same for us because we're animals, but you get the picture. And of course, foliage there. So foliar pathogens usually are more aesthetic based in case they get really, really bad. In the case of this one, I picked this. This tree didn't die from a foliar pathogen. It was just the stark photo. Foliar pathogens you'll see on your Colorado blue spruces, such as Rhizospora, like your needle casts, and they can absolutely wipe out a tree. But your more common one that I think everybody would be familiar with in the urban environment is tar spot on maples. And those are those big ugly black spots that you see start to form right now. They don't actually affect the health of the tree that much. If you think about each leaf as a solar cell, those black spots, they're like dust on the solar cell, and they come in so late in the season. They actually exist for the entirety of the season, but they affect the plant very little. And when they start to reproduce at the end of the season, it's not a very big deal, right? So that's what I would consider an aesthetic problem. It's not a physiological problem. It's not a structural problem. It does affect the tree's ability to produce sugars, but very minimally. Some of them, like Diplodia tiplight, otherwise known as Spheropsis sapinea, you'll see that on your black pines, right? Your Pinus nigra, or your Austrian pines, quote unquote. And those ones they can wipe out a tree over the course of many years. And when I say many, I'm talking like 10, 15 years. Okay, moving right along. Where else are you gonna find them? Roots, right? So what are some of your symptoms? The growth is gonna be reduced. You're gonna see dieback in the canopy, right? Those are all your symptoms, not your signs. Look for construction damage. I see this a lot in the municipal environment and sometimes with my residential clients who haven't been my clients for very long. They'll be like, oh, my tree's dying. And they've cut a new driveway and taken half of the roots out. And it's like, well, the tree's lost half of the roots. And now you've got a fungus that's come in and taken advantage of all of those massive wounds and is wearing away at the remainder of the roots that exist, which can be lead to structural problems as well as physiological problems. Armillaria right there. So that's one that is a root pathogen and a decay fungus, right? And so decay can happen a number of different ways. It can enter through a taproot that dies, and that's a part of the normal process. Or it can happen through poor pruning cuts. It can happen through storm damage. Anytime the bark is impacted, you are exposing a tree to decay. Anytime you prune a tree, you are exposing it to decay. So beware, for those of you out there who prune your trees, the best thing to do is to prune them when they're young and to do more small cuts and less big cuts. So if I can do 10 cuts on a tree that are 3 centimeters a piece in diameter, or 1 cut on a tree that's 30 centimeters in diameter, 10 times out of 10, I will take 10 smaller cuts because it's far easier and there's a far greater surface area so that the tree can respond and not heal, but seal over those wounds. And if you have more questions about that, I'm very happy to talk about it. Decay, very significant for safety. So if you're dealing with trees in the urban environment, which is exactly what we're dealing with, typically we have to prioritize risk over the physiological health of the tree. And that's why we have to look at a tree, assess the decay. And for me as a tree risk assessor, I very much an advocate for trees, but priority is always myself, the public, my clients, right? Not particularly in that order. Sometimes I put myself at the bottom of that list, but you understand the point that I'm making. Decay is very important for managing a tree because if there's extensive decay, sometimes it means the tree needs to come down. If there's minimal decay, sometimes it means you can just manage the tree and keep it going. But it's a case-by-case scenario. Okay. So important decay fruiting bodies. You got your Ganoderma, your Phalinus, and your Climacodon. So these are ones that you will see in the urban environment relatively often. Your Phalinus is neat because it looks a bit like a rock. That's a side profile pic, and there should be another one in here that's a little bit more detailed and shows what it looks like in the tree. But these are ones that you should see, and if you see them, doesn't necessarily mean the tree needs to come down, right? Keep that in mind. It just means that you should earmark it, keep it, and watch it, right? Because there could be, it could get in there through physical impact, right? Whether it's a car driving into it, that's a really common one we see at the side, on street sides, right? Sometimes we see that in our driveway, you know, it's a Christmas party and someone's not paying attention, it's late, it's snowing, they're backing up and they back into your tree and that's not a big deal. 10 years down the line though, it will be. Different species manifest in different locations due to the chemical composition of the wood. So Ganoderma will never appear in the canopy of the tree. It will always appear near the base, near the roots, and a little bit near like what I would call the lowest scaffold branches. So you're not going to see the Ganoderma anywhere near 10 or 20 feet. Pardon me, anywhere beyond 10 feet. Once you're getting to 20 feet, that's pretty rare to see Ganoderma. You will sometimes, but not often. Because this is one of the things that we won't go into too much of the biochemistry here today, but wood is not uniform. The wood at the base of the tree has a different chemical composition than the wood at the top of the tree. Often, not always, but often. And so fungi know this, and they prefer, depending on the type of species, certain types of wood. Some decay fungi actually prefer eating in the canopy of the tree. They prefer breaking down lateral limbs and the top of the tree versus other ones prefer the lower or the central parts of the tree. Okay, so Ganoderma, right? It decays both cellulose and lignin. I would call it a preferential lignin decayer. Once again, we're getting really far into the biomechanics, and I don't want to get too far into the weeds there. You'll lose loss of strength after inoculation. The perennial fruiting body can live up to 10 years, and there are columns of decay, right? So it doesn't just exist where the fruiting bodies are, and this is what's important to keep in mind. Where the fruiting bodies are gives you an idea of where the fungus is, but it doesn't tell you the whole picture, right? It's just a snapshot, because remember, those hyphae, those mycelia, they live in the substrate and they move. So the mycelia can move all the way up. This is a hemlock, so this is a Ganoderma sugei, not a Ganoderma applanatum, that's different, but you can see the margin, which is very indicative of Ganoderma, and those beautiful orange colors. This is in Algonquin Park. It's quite unfortunate too, you know, you talk about how people take things. Anytime I'm on this trail, this is the hemlock trail, the fruiting bodies are removed. I know where all the fruiting bodies are because I'm a nerd, but they're often removed. I was there just a couple weeks ago and every single one of them were gone. Bearing that in mind, the fungus will exist all the way through there, and the longer those fruiting bodies exist, the more dollars to donuts, the more you can bet that wood is getting more and more decayed. All right, so Phalinus, there you go. So you see how it's that nice black horseshoe shaped looking fruiting body, and you look behind it and how gnarly all of that is. That's it breaking down, and it can actually cause its own cancers as well. So it's a white rot, which means it breaks down the lignin, which is the compressive strength of the tree. We won't get too far into that, like I said. And it's a perennial fruiting body. So if you see it, you can actually get an idea from from those little, I hesitate to call them rings, but for lack of a better word, I will call them that. And you can see it's quite a large fruiting body, 25 centimeters across and probably about 15 centimeters from top to bottom. And they live long enough that you get moss growing on them. And the nice part about that for a professional like myself, or even for citizen scientists such as yourselves in the HNC, you can look at it and get an idea and say, oh, okay, this thing's been living here for a long time. If you remove it, you're not removing the fungus, but you are removing a sign of the fungus and part of the fungus, the spore producing factory. I'm still on the fence on whether or not it's a good thing to remove it, partly because for myself as a professional, when I walk on site, I want to actually be able to look at that and get an idea of how long that's been there, because it helps inform me when it comes to my tree management plan. Because in some circumstances, I might look at it and say, oh, this has been here for 10 bloody years. Probably a good idea to reduce the tree or to remove the tree. Whereas I can walk in there and say, oh, that's only been there for a year. Tree looks fairly good. Okay, maybe we can just leave it and revisit in 3 or 4 years. There's another example of a Phalinus on an ironwood. Surprise, surprise, my business is ironwood, so I love those trees. They're one of my favorites in the world. And Phalinus are the only fruiting bodies that I happen to ever find on ironwoods, other than Inonotus obliquus, which is a super straight. All right, Climacodon, white rot. So it also affects the compression strength of the tree. And you can actually get a bit of an idea, it's just a bit of a glimpse here. If you look at the base of the sugar, it goes in, it kind of flutes out a little bit, and then it goes back in. That sort of fluting we call a bottle butt, and that's actually indicative of the extent of decay. So I can walk onto a site, see what the tree, how the tree is expressing itself, the structure, and it gives me an idea of how advanced that decay is. So one of the important things to note about this fruiting body, it's got beautiful little teeth. And now it's a very fleshy one, so it doesn't last very long. It's not like the Ganoderma, which is as woody as it is. You know, I can take it out, I can put it with my samples, and I can use it as a teaching tool. Not so with Climacodon. Climacodon, unfortunately, only lives for about, less than a season, about 4 or 5 weeks after it fruits. This is the time of year where you will start to see it come out. Sometimes you'll be lucky to see it after a rain in July. But otherwise, who here has actually seen this before? Because this is one that I'm always amazed at. Wow, okay, so 8 people. That's pretty impressive to say the least, because you got to catch it at the right time, right? It's not like the other ones. And those teeth are actually where the spores are produced. So that's a whole other type of spore producing layer. And it does produce a lot of decay, right? 4 meters above and 1.5 meters below. There's a really nice one on a sugar maple. I hope you guys appreciate my artistic eye. All right, Polyporus squamosus. This is your dryad saddle. This is relatively common. A lot of people see this one. Same song and dance as the other ones. It affects the compression strength. It doesn't live for very long. Like, it'll produce same sort of thing. It's a seasonal fruiting body, and you'll see it all through the season. But if you come at it and find it a year after it's fruited, you can actually still find parts of it, because what it'll do after it sends out all of its spores is it will shrivel up and turn black and hard. It smells like fish. It's super gross, but it does let you know that there is that decay there. And it's one of those ones that I would consider a yellow flag. It's not a big deal. It's something that can be managed as long as it's not ignored. All right. So the interesting thing about cankers. So we've talked a little bit about cankers, and we talked about decay. This tree actually was removed this year because the decay had become so extensive. But butternut canker is an introduced species. The canker has come in and it's completely ravaged the cambium. Now, interestingly enough, the tree was still vigorous. Physiologically, it was still healthy, but the decay was so extensive, and it was extensive because the cambium pathogen, the fungus that destroyed the cambium, completely exposed the interior wood to decay. Not good, to say the very least. Not good at all. So that's something that can really shorten the life of a tree. And butternut is one of those endangered species that we have in Ontario that we're really trying our best to actually manage. And it's important to understand that sometimes, even if it's an endangered species, the best way to manage it is to remove it, because it's a significant hazard. And this was a massive, massive butternut. They needed a crane to take it down. And it was in between 4 or 5 different houses. There was no safe way for this tree to fall over. So took it to the ground safely. There's some of the smaller fruiting bodies, which we talked about a little bit. I'm gonna see if I've got one here, which I believe I do. So we're gonna just flip really quickly to the microscope again. So I'm gonna do a new share to the Micro Capture Pro. Nice. Okay. So this is on a Norway maple limb that's been ravaged by a canker. So you can see all of those little black dots right there. Those are all fungal fruiting bodies that have destroyed the cambium, which we would consider a canker, right? It's a cambial pathogen. Go in and out, very cool stuff. So flip to the other side. And so you've got, you can see on one side, this is all healthy tissue. And then dun, dun, dun. Terrible and ugly. It is full of fungus. And these are all little disease-making factories, right? So this is what I mean when I say there is a lot going on your tree out front. This was just a client's tree that we'd come out to prune for the first time, a little Norway maple. And we were taking it off, and I said to my apprentice, oh man, Ryan, grab that. Like, this is awesome. And then I spent 45 minutes that night looking at this thing under a microscope because it's fascinating. And this is why when I say we can do a fungus walk any time of year in the urban environment or in the natural environment, I mean it, because this stuff extends far beyond your typical mushrooms. And like I was saying earlier, this is an ascomycota, right? So this is part of your invertebrates. All right, so moving back to the slideshow. We're just about done here, folks. I really appreciate everybody sticking around and paying attention. Mycorrhizal fungi, right? So we've talked a little bit about that. So these are your symbiotic ones. They aid in the uptake of phosphorus, and success is based on species. So you want to have a good relationship. And if we're talking about trees in the urban environment, trees need good soil, and so do the mycorrhizal fungi. And this is another chicken and the egg thing. What do you do to improve the case of trees in the urban environment? We need to improve the soil for the trees in the urban environment. We need to improve the biota that's in that soil, because it's all part of the same thing. And the urban environment is so damn harsh. It's really harsh. You got salt half of the year. People don't water their trees. They get hammered by sun. They get hammered by snow. If they're at the side of the road and they're getting hammered with salt and then snow, it's just really, really tough. And then you have the soil compaction issues. And if you're in a new subdivision, you can bet dollars to donuts, when they started building that subdivision, they scraped off all of that high quality topsoil and sold it to someone. And now all you really have is the B horizon, right? You have the runner up of soil to grow your trees and your mushrooms in. Not ideal. Neat thing about it though, when it comes to the mycorrhizal fungi, you can actually see where the roots, that's a white pine right there, and conifers are really well known for having mycorrhizal associations. You can actually see where the roots of that pine are. Kind of like I was saying earlier, when you pull up the substrate, look at what the mushroom is growing out of, right? So there's a bolete here, you got a chanterelle, and then this little fellow right there, that's your Amanita muscaria. So if you're familiar with Super Mario Brothers, or if you are familiar with the Siberian tribes people of the Irkutsk region, some of you may be, that's a very important shamanic mushroom. Highly hallucinogenic and poisonous, but really it's the dose that makes the poison, right? So moving right along, interesting thing, and I had mentioned this at the beginning, these are both suolus, picked one day apart. So I was on this site assessing trees for risk, and this was on Monday and that was on Tuesday. That's how radically different they can appear. And you can see there's pores there. So pore mushrooms that grow out of the ground that have pores, we call those boletes, B-O-L-E-T-E-S. There's some more glamour shots of the Amanita muscaria. So these are really, really stunning mushrooms to photograph and to find if you ever find them. And one of the ways that you can pick out your Amanitas is if you look at the bottom, they've got that little cup. It's called a volva, V-O-L-V-A. And it's kind of like an egg that they sprout out of. And that's the same with the Amanita virosa. So Amanitas, interestingly enough, the entire genus, about 35 million years ago, gave up their ability to break down wood and decay in exchange for their ability to trade nutrients with trees. And there's actually some really interesting research on that out there. And I encourage you to go and seek out those papers, because Amanita as a genus of mushroom, or as a genus of fungi, absolutely fascinating. Absolutely fascinating. I strongly encourage. Okay. This is my bad Titanic joke. Okay, so what exactly was that terrible joke about? So when we're dealing with trees, trees don't have the ability to heal all they have the ability to do is seal. Thus Titanic. What was one of the biggest problems with Titanic? They hit an iceberg, yes. But other than that, the floodgates, they had big problems with their floodgates because they didn't close them fast enough. And so after they hit the iceberg, my wife is shaking her head at me because Titanic is her favorite movie. After they hit the iceberg, they didn't respond fast enough. Because they didn't respond fast enough, it's one of the reasons the ship sank. When it comes to trees, it's the same sort of issue. There are good compartmentalizers and there are bad compartmentalizers. Trees that compartmentalize well, ironwoods, oaks, some of your maples, basswoods, they can have decay move in and they can stop that decay from actually moving throughout the wood, right? So if you saw that, or if you remember earlier from when I was showing you the locust, there was part of that locust that looked beautiful and creamy and white and healthy, and then there was the other part that looked like it had been shot through with a shotgun. That is because part of that locust had actually compartmentalized really well, and the other part hadn't compartmentalized at all because it had been infected by the fungus before the tree responded properly, which is one of the reasons why the tree had to come down. Now, if you look at this Norway maple on the side, you can actually see how far the fungus is moving through, but it's actually moving up and down more than it is moving out, and that's part of the tree's compartmentalization system. This is why you want to prune trees well, and this is why we want to protect trees from construction in the urban environment. It's very, very important when we're dealing with trees in the urban environment, when we're dealing with the urban forest, to mitigate the risk of exposure to decay and to really beware of what trees are good compartmentalizers and what trees are poor compartmentalizers, because that helps us make informed decisions, not only when it comes to planting, but when it comes to managing risk. Decay affects wood in a variety of different ways. And because we're getting to the end of their day, and I'm sure everybody's kind of running to the end of the energy to pay attention, I don't want to keep pushing this, but we've got lignin and we've got cellulose. And so understanding the difference between lignin and cellulose is going to really help you understand what kind of decay you're dealing with and how decay actually affects the wood. Because when decay is in there, it's eating different parts of the wood, right? So as I was talking about Ganoderma earlier, how it primarily breaks down lignin and then it breaks down cellulose, you can actually get an idea of what it's breaking down based on the color of the decay. So when you're out and you're picking up decay that's really, really fibrous and soft, that's your white rot. So it's leaving the cellulose behind, and there's some tensile strength that actually remains in that. When you're looking at brown rot, that's the sort of stuff that crumbles. So chicken of the woods, one of the most iconic that we haven't talked about today because we might talk about it a little bit later, I'm not sure. That's one that really is something to watch. Brown rot is oftentimes a red flag. So there's what your white rot looks like. So, and once again, I understand these are misnomers and we can really get into the weeds on that, so I won't. But you see how it's peeling off like a flake of paper. That's your white rot. Very, very simple to pick out. Soft. And there's another example of those floodgates there. So this is a Eucalyptus polyanthemus, otherwise known as a red box. And you can see eucalyptus trees are excellent compartmentalizers. So this is an old branch wound right there. And we go into the next step and look at how well it's actually compartmentalized a lot of that decay. So nothing else out here has been affected by it. Unfortunately, this tree needed to come down because, well, they were building a parking lot over Paradise. I'm sure there's a song there somewhere. But, you know, in the process, I got some really interesting photos. And you can see up close how it's actually moving through. So the other side right there, just it really arrested it. And keeping in mind that that's, you know, you're thinking of a 3D image and we're only getting a snapshot of that 3D image. It's really hard to peel the whole thing apart, especially on a job site. But you can see what good compartmentalization looks like, because that decay got in there and it really didn't get that far into the tree. And part of that is because the wound where the branch was taken off years prior was rather small. All right. So how are we managing trees? What are the risks and rewards of this? Right? So if we're managing trees and we're thinking about trees in the urban environment, we're thinking about fungi and how they're affecting trees. What are we doing about something like this? So at the right-hand side, you've got a playground, and on the left-hand side, you've got a really nice elm tree that has right at the top a little fruiting body there, right? So what do you do? Now in this circumstance, this is a particularly nice tree. It was fairly large for that environment. Obviously a little bit of construction going on nearby, but no reason to cut the tree down. And so we ended up just reducing that limb. You can close down the whole daycare center, which is kind of unrealistic, or alternatively, you can just prune the part that has the decay back and accept that maybe there's still a little bit of risk there, but you're not going to send the kids out to play out in the storm anyway, right? Same song and dance here. Whoop. Going back, these are those poplars from Russia, different location, but same song and dance. So this is a couple years on. So when the leaves are on, right? I went to Russia in winter. Crazy, I know. Because it's not cold enough in Canada. But you look at this and you think about when the leaves are on the trees, they would absolutely fill their role, right? They would be nice and green. They would be good for managing a little bit of the sunlight. And if you notice, there's almost only poplars there. That's the only tree that they have to manage. So you do what you can with what you got. Here is another nice example of a hydro prune. Personally, for me, I think the better opportunity or the better response to this is just to not plant large trees under hydro lines. Because then you have to do this, and as amazing as that is from an infrastructure management perspective and from a pruning perspective, it's only a matter of time before that tree either has to come down prematurely because of those lines, or the wind takes it down and those lines go out with it, and then you have another problem on your hands. This one here is what I was talking about that school earlier. So this is actually from where I used to live in the Dandenongs in the state of Victoria in Australia. Beautiful place. This is the house that was right around, or this is the school right around the corner from where I lived. As you can tell, this is a tree that had fallen over. And there you go. What happened exactly? Well, tree fell on the school, fortunately on a Sunday. This is a Eucalyptus regnans. The tree was about 200 feet tall. Massive, massive trees. If you ever have a chance to look at Eucalyptus regnans, largest flowering tree in the world, the largest tree species in the Southern Hemisphere, overall the third largest tree species on the entire planet. And arguably, if they hadn't been cut down so much and deforested, they might actually be the tallest tree species on record. But that being said, this is an example of poor planning. So you can't see it here, but I actually did a little bit of digging, and there was a major structural root that had been damaged while digging a post hole for this nice little fence. So when it comes to construction, are you planning your construction projects around your trees? You don't have to, but if you want the trees to last, you might want to consider that. Or if you want your trees to last and to not fall on your things, you might want to consider the costs and the risks and the benefits. Here's another example. This is in Tasmania of a beautiful Phoenix canariensis palm. Absolutely stunning trees. If you ever have a chance to appreciate one in person, highly recommend it. They actually built the extension of this place around the tree. So I was driving down the street and I saw it. And of course I ran 4 people off the road to park and run in as soon as I could because it was fascinating to me. And I said to the guy, like, what happened? Why did you do this? Like, bring me up to speed. And I guess what had happened was this was a very iconic tree. People were parking their bikes in before they went into the bike shop, and they wanted to extend and get more room and at the same time protect the tree a little bit because everybody was putting their bikes against the tree and it was starting to wear on it, right? Little by little, like water on a stone. And so they built the extension around this tree. I could talk to you for hours on how they did it. It's a fascinating project, but essentially what he said at the end of the day was it was the best decision they made because it brought more people in to look at the tree than they had ever had to look at bikes. And as a result, they opened up a cafe in the bike shop that ends up making more money than the bike shop actually. Fascinating, bloody fascinating. So there are good ways to make money off of your projects when you're protecting your trees. Yes, it might take an initial investment, but there's a good ROI. as can be demonstrated right here. So we want to solve our problems, right? We want to solve tree problems before they become problems. And one of the ways to do it is to understand what we're dealing with. And one of the biggest issues that I find with trees falling on homes is benign neglect. So it's people that have the wrong tree in the wrong place, or they have an instance of a tree that was damaged in construction many years ago and they were unaware of it, and then it ends up being somebody's problem. So in this case, this is a bitternut hickory in Hamilton. This fell in 2013 in a major storm on July 23rd. And it took 14 hours to get that bitternut off of the house. Fortunately, there were only 4 holes in the roof and we didn't damage anything else taking it down. And you can see in the top right-hand side, the size of that tree, quite large. And right here as well, what it looked like afterwards. So we did what I would say a damn fine job getting rid of that. But I took a cutting of it home just to see, and sure enough, that is a foliota, fruiting body. So that's the bitternut hickory there. I left it there for a year. I waited and waited and waited for it to reproduce, and sure enough, it did. So you can see all the little hyphae coming out of the wood and the fruiting body coming out of the center. Now, interestingly enough, this whole subdivision was built about 14 years prior, and that bitternut was part of the fence row that used to exist when that place was a farm. And what happened? Well, they put in a real fence up, and sure enough, like the eucalyptus on the school, or like the locust tree that I had to take down because of my client putting the deck in, the tree failed because of decay. And the decay got in there because of poor construction and poor planning. And this is a decision that nobody that was living in that house made, right? And it could have been easily prevented if it was done properly and if the planning was done with the help of an arborist. So there you go. So why is any of this useful? Because we want to know what decay is. We want to educate ourselves, right? And it's, yes, a little bit of information can be worse than no information. We need to start somewhere, right? We don't need to be afraid of trees, but it's good to be aware of trees. And sometimes that can mean just putting up signage to say, if it's really windy, don't go out, right? If it's 40-kilometer-an-hour winds, it's just not feasible to walk in the forest. I don't, and I say to my wife, like, I'm not allowed to get hit by a tree. I really don't, that's not the way I want to go. And I know too much. So in those circumstances, if it's really windy, it's just better to let Mother Nature take her course. Construction is a major entry point of decay in the urban environment, and that's something that we really need to manage. In the city of Burlington now, we have a tree protection bylaw. So if you're doing any construction on your property at all, you need to have an arborist report and you need to have a tree protection plan. Because one of the things that we found out is when you don't have tree protection plans, trees fail, like we've seen today, and sometimes those can be municipal trees, and that costs the taxpayer far more money in the management and the replacement of those public assets, really. Because if you get a really nice mature tree on a street and it's a public tree, and something happens to shorten the life of that tree, it's going to takes 100 years to grow a 100-year-old tree, right? And if it takes a couple days to cut it down, and it takes one bad construction project to shorten the life of that 100-year-old tree, right? So there are things that we can do to actually extend the life of trees in the urban environment, bearing in mind all of the other challenges, all of the fungi, all of the crappy soil, all of the insects out there, all of that. There are still ways that we can actually extend the life of our trees. And we need to look at the possibility of mycorrhizae and other soil biota improving the quality of urban soils. So finally, in order to properly manage and improve the quality of life for our urban forests, we need to be aware of the connections between trees and the species that live in and on them. And you folks are exactly the kind of people who can do that. You've got a great knowledge and a great skill set when it comes to communicating with the public, managing your own trees, educating yourselves, your friends, and your family. And so you are part of this. You're just as important as anybody else in actually improving our urban forests. So with all of that, thank you very much. If you want any further reading, a good introductory book, Mushrooms of Ontario and Eastern Canada, Fungal Strategies of Wood Decay in Trees, is a really good advanced book for those of you out there who are practitioners in this sort of thing. I encourage you to look at strongtree.ca, so that's Ironwood Arboricultural Solutions website. we're trying to selectively grow our clientele of people who care about trees, who want to manage trees on their property and understand the value of high quality arboricultural work. So that's everything from managing trees on your property to planting trees, pruning, removing, etc. Unfortunately, we don't operate in Burlington because of my position with the municipality there, but we have, we're taking on select clients right now in Hamilton and in Brant County and in Wellington County. Online also, check out treerot.com. That gives you a little bit more information on wood decay fungi on living trees. That's Chris Luley's website, who's a great PhD out of the States. And then the Science of Arboriculture podcast through ISA, excellent podcast if you ever have a chance to look at it. And finally, if you have any questions, I'm happy to go through some now, but also there's my email at the bottom, ironwood@arboristabroad.com. And very happy to field any questions that you guys have. Really happy that you've been here, and we've had some great numbers. So I'm very excited about that. Okay, so I'm gonna pass it over. I'm not quite sure how to, but I'd like to pass it over to, oh boy, Chris or Lou.
Point to my name, and you'll see a couple of buttons come up and says more. Click on more, and then
anyone there? Am I just by myself right now?
No, can you hear me? Kyle?
There we go. Hey, sorry.
Okay,
Thing. All right.
okay, good. Okay, so you point to my name on the panelist list, click on more, and make me the host.
No Facebook, no. Oh, this is so embarrassing. I'm a millennial. I should know this stuff. Record to the cloud, chat.
On the participants panel.
Yeah. Oh, there we go.
On the panelist panel, click on more when you point to my name on the right.
Brilliant. You are the host.
Switch it over. Okay, thanks. Thank you so much for Kyle. That was great. Just fascinating, really stuff that I had no idea about, and I'm sure a lot of people were really curious to find out. And thank you for everybody showing up. There were 63 in total tonight, so that's pretty good for a video or webinar on this. There are a couple of questions
Chris,
here. Yes.
Chris, there's actually 3 questions under the Q&A.
There are.
So Kyle, I think you can probably click on the Q&A and see those questions and just unmute yourself if you want to answer them.
Okay, Q&A, I see 3. Brilliant. Why do they tie the trees like that? What is a spore print and what can be done to prevent butternut canker? Oh boy, okay. So we're gonna go to butternut canker first. Nothing can be done to prevent butternut canker. Because it's an invasive pathogen and it's particularly virulent, you can't do a lot. Beware about pruning your trees. I would say if you're pruning, prune in the dead of winter when the fruiting bodies typically aren't, they can still spread their spores, but they're not spreading them actively. What is a spore print? Spore print is something that you'll use to actually identify the trees, or pardon me, identify the fungi. So spore print is something like if I take a cutting of a mushroom and I put it on a piece of paper, if you leave it there for a certain amount of time, what's actually going to happen is the gills of the fungus will actually reduce the spore or release the spores. The spores will sit on the paper, and if you take that off after a period of time, like 30 minutes to an hour, you can get a really good idea of what the spores look like, because they can be white, they can be pink, they can be green, they can be brown, they can be red. And if you're looking at any sort of field guide, most will give you an indicator of like spore print yellow, spore print red, or spore print blue. Not that I don't think there are blue spore prints, but anyway, neither here nor there. I hope that makes sense. If it doesn't, fire me another question. Why do they tie trees like that? So if you're talking about, and if Cryptic FI is asking me that, if you're talking about why do people tie trees when they plant them, typically the reason we'll plant trees, or trees will be tied when they're planted, is to keep them stable. Now, it's a little bit of an outdated technique. In the last 5 or 6 years, people have stopped doing that because we found that it's like training wheels. If you never take training wheels off a kid, they don't learn how to ride the bike properly, right? And so as a result, if you leave a tree tied too long, and this is one of my experiences, either nobody ever takes those ties off, and then the tree grows into the tie, it girdles the tree, and it can actually shorten the life of the tree. Or, even worse, the tree just doesn't learn to stand up on its own 2 feet. And then it just falls over in a windstorm. And that's part of the purpose of having those ties, is to prevent that. Now, typically, if the tree is planted properly, that shouldn't happen. So you shouldn't really need ties. Sometimes you might need 1 stake, but not more than that. How does someone become an arborist? So arboriculture is an interesting trade because there's no real government regulation, like with carpentry, plumbing, hairdressing, or an electrician even. There's the Ontario Ministry College of Training and Universities. You can do an MTCU apprenticeship, which is anywhere from 4,000 to 6,000 hours. And I would say that's the more practical side of arboriculture. So that's the climbing the trees, operating chainsaws, understanding how wood reacts. And then there's the ISA certification. So the ISA is the International Society of Arboriculture. I'm on the education committee for the ISA. I'm actually doing an ISA webinar next week, which is going to be a lot more in depth than this one has been. And with the ISA, you need about 5 years of experience in the green industry before you challenge the test. And then you just have to write the test, and then you can get your ticket. And as far as I'm concerned personally, I think that we could raise the bar a little bit, because there's a lot of different things we can do to improve our industry. And part of it is having higher expectations for our professionals, because some people out there, you can become an arborist and have never operated a chainsaw or climbed a tree. Doesn't mean you're not a good arborist, but there's a degree of education and experience involved in that. Okay, oh, Phil Davis, great question there. Almost all of my trees have split bark, usually on the southwest side. How dangerous is split bark to the tree? Well, without seeing the tree, I can't really comment on how dangerous it is, but you are talking about a phenomenon that we refer to often in the industry as Southwest syndrome, which is exactly what it sounds like. It's not exclusive to the Southwest. You can get it on the north side of the tree, depending on what's around. But what it typically is, or what it is caused by, is the movement of sap in your warmer, when spring is coming along. So your warmer days in March with your cool nights, the warm days, the sun will heat up the southwest side of the tree. That will get the sap moving. The sap will move its way to the top, start filling those buds full of sugars. And then after the sun goes down, the temperature drops rapidly, and it drops faster than the sap can actually get down to the bottom of the tree. And in the process, it will actually freeze at a location on the tree and cause a crack. That's your Southwest syndrome. And year after year after year, that happens. And it's difficult to mitigate, because once it happens, it cracks the actual underside, like the interior of the wood, and it can cause some significant problems down the track, but it's not a reason to cut a tree down. Let me put it that way. Oftentimes, it's not a reason to cut a tree down, depending on the size and the targets. But bearing that in mind, it's something to watch, because sometimes it can be really bad, sometimes it can be quite simple, and the remedy in some circumstances can be to paint the tree white, believe it or not, because that actually mitigates some of the temperature. In other cases, it can be just a case of your home is white or really bright and facing south, and all of your trees are right in front of that white wall. And so it's going to heat up really, really fast, and then it's going to cool down really, really fast. And so that's a problem that once you have it in a tree, it's there permanently. It's going to happen every year, usually, but it just becomes how should I put it? It just becomes a problem. Really, at the end of the day, it becomes a problem that you have to deal with. And it's kind of like having a bad knee. You know, you got a bad knee, it's there. You just kind of have to work with it. It's not going to necessarily shorten your life, but it means you're not going to get to play, I don't know, high-a-lie or do bobsledding anymore, or what have you. Did I answer all of the questions here? Why do they tie trees like that? What is a spore print? What can be done to prevent butternut canker? Just an interesting note on butternut canker too. Butternut canker isn't the only host. Butternuts, pardon me, juglans cinerea aren't the only host of butternut canker. Walnuts, black walnuts are also a host of butternut canker. So you can have a walnut that is infected with butternut canker that is in relatively good shape, and unfortunately, it is just a disease factory for butternuts. And so what it does is it produces butternut cankers, and it's not, the butternut canker in the black walnut isn't really shortening the life of the walnut, but it is producing more and more and more fruiting bodies to spread to the butternut. Okay, what work did I do in Melbourne? So I worked in the ranges for a little while for the Arthur Rylings Institute, specifically for a arboricultural firm there, doing leadbeater's possums research. So we would go into the forest, set up cameras and camera traps, bait traps, to see if there were leadbeater's possums. Because if there were possums there, we would actually prevent any logging in those specific logging coops. So every time you'd see a leadbeater or proof of a leadbeater's possum, you could set up a 12-hectare exclusion zone from logging. And so we would go in usually just a few weeks to months before they actually had a logging operation to try and identify whether or not the leadbeaters were going to be affected. So that was one part of what I did. And then I worked for a residential tree company for the other half of it, which was one of the reasons I wanted to go. So as a professional, I wanted to do what you would call a journeyman year. So I had finished all of my tickets and my experience, and I decided to go down there. I've always dreamed about the Dandenongs and about Eucalyptus regnans, the biggest, most beautiful, fantastic trees in the world, as far as I'm concerned. And so going down there to climb eucalyptus trees and challenge my botanical knowledge and learn a little bit about how the industry is practiced, how the profession is practiced in another part of the world. Okay, so we're at about 9:21. I'd be happy to answer one more question, but if everybody is satisfied, I am as well. And I really appreciate everybody actually taking the time to reach out and connect. It was a pleasure. And you know, it's always tough to see where people are at when dealing with something like Zoom, because I can't feel you guys out. But it was nice to see that people were actually raising their hands, which is brilliant.
That was a big help. That was great. Thank you so much, Kyle. It was fabulous. And also, if you do have any questions, as Kyle mentioned earlier, you have his website or his email. You can always field a question that way.